We have had the introduction to the kingdom fungi. We had talked the phylums in which it was classified further to 4 phylums, and we had also discussed the basis on which that phylum was classified. What we had not discussed was about reproduction in fungi in great detail. So, we are going to study about reproduction of fungi now. You know that there are 2 methods of reproduction. One is asexual and other is sexual method of reproduction. In terms of sexual method, we have a detailed process, but the asexual method is very simple, that either it would be by the fragmentation process where a fungal hypha would separate and it will form a new mycelium, or it would be by budding. A bud would be formed, bud separates and the mycelium would be given rise to, and the third way is asexual spores. When we talk about asexual spores in case of kingdom fungi, you have to remember that the 2 types of spores, that is zygospores and conidiospores. These spores are very important when we talk about these what we call fungi, the asexual spores I am talking about. And in terms of sexual reproduction, we have a detailed process, and on basis of that sexual reproduction, there is a classification that is being made. There are 4 types of fungi. Now, first of all, let us see the life cycle of fungi, what it is all about. You know, fungi is again a thalloid organization. What is a thalloid organization? That means there would be clustering of cells, but those cells would not be working as an entity as total. I hope I am clear that the thalloid organization is one such organization, where if I am talking about fungal mycelium, you would see that I am talking about so many cells, that whether they are septate or they are not aseptate, but at the same time those cells would be individual entities. If I see that fungus, that would be one organism to me, visible to me or any onlooker, but it would be, each cell would be together, working together, but not as a tissue. So, we do not have that level, that organ level is not there. In fact, the tissues are also not differentiated well, so that we can call them a tissue. So, this is also a thallus-like organization. And not to forget, fungi are somewhat related to the slime molds, and we did talk about cellular and acellular structures in the case of slime molds. Now, what we are going to see is the life cycle. And as I tell you, because they are very simple organisms, they are going to have the simplest way of life cycle. Either they would be reproducing asexually, or it would be sexual reproduction method, and there would be alternation between the haploid and diploid phase in the life cycle. So, we have to pay attention to that phase as well. You know that the fungal body is haploid. So, what we see over here is that the mycelium is haploid. We see the fungal mycelium. Now, if it has to undergo asexual reproduction, there would be mitosis in these cells, mycelial cells. Some of the cells would undergo mitosis, there would be differentiation. Mitosis would take place, and that would lead to formation of asexual spores. Like if I say conidiospores, if I say other spores, asexual spores like sporingiophores and ascospores are sexual ones. Just remember that the spores which are being formed, they would be asexual spores. There is germination of these spores, and again the mycelium is given rise to. Talking about the sexual reproduction, there are 2 stages in the sexual reproduction of fungi. The mycelium would undergo first stage, that is known as plasmogamy, and the second stage would be karyogamy, and in the meantime there would be a condition known as dikaryon. Very important thing you have to remember, a dikaryon is being formed, and what is that dikaryon? Let us come to that as well. See, this plasmogamy is what it has been described over here, haploid cells from 2 different mycelium, which would be considered as plus and negative strain. We will come to that. Haploid cells from 2 different mycelia, they fuse to form a heterokaryotic cell. Now, what is a heterokaryotic cell? That is a dikaryon. As you can see over here, this supposedly this is one cell's nucleus and this is one cell's nucleus. Because the plasmogamy has taken place, there is a heterokaryotic stage where both the nuclei are present inside a single cell. That heterokaryotic stage, it symbolizes that plasmogamy has taken place. Okay. Once the plasmogamy has taken place, then it is to be followed by karyogamy. Now, gamy word is fusion and karyo is for nucleus. We have discussed about it. So, there would be fusion of nuclei, two nuclei which were a heterokaryotic stage was having the two different nuclei of two different strains of two different fungal mycelium. They are going to undergo a fusion. The nuclei fuse to form a diploid zygote. So, till here we were seeing that this part was haploid. What we are going to see over here is the diploid stage. These two nuclei has fused to form the zygote. Now, when this zygote is diploid, because the main body of the organism is haploid, it has to undergo meiosis. When the meiosis would take place and the successive mitosis can also take place, the meiosis has to take place so that this diploid zygote gets converted to haploid spores which can germinate, and these haploid spores are sexual spores, sorry, pardon me, it won't be asexual, these are sexual spores. On the basis of these asexual and sexual spores, we have classified fungi into further 4 divisions basically. Now, let us see what is this basically. This is asexual reproduction in fungi. As you can see, this is a bread, if you can make it out, and you have seen that whenever the bread rots, there is fungal growth on the bread that is known as bread mold, and that has the typical name rhizopus. All of you know that. That greenish development, rotting of bread, greenish black rotting of bread, if it is seen under microscope, it would be this. This part, this looks like some plants are grown, but it is not that. This is microscopic depiction. You see the fungal hyphae over here, the mycelium, and on this you see sporangium, lot of sporangium which are developing. Inside the sporangium, there is mitosis and development of sporangiospores, the spores which are asexual spores. These spores, they are going to germinate and give rise to the next fungal mycelium. So, this is the simplest way of asexual reproduction. As in the case of penicillium, they do not have sporangia over there. Okay, penicillium is a very famous fungi responsible for development of penicillin, which is an antibiotic, and later on it unfolded many stages of antibiotic development. This penicillium is an ascomycetes. It has conidiospores. Okay, like we have sporangia over here, over there we are going to have conidium, and inside the conidium there would be conidiospores, or we can say conidia. Conidia is the exact word, not conidiospores. Conidiospores could also be used. There are many conidia which develop and then they germinate and give rise to fungi. Next is the sexual reproduction in fungi. As we had discussed earlier, the gametes are formed, whatever gametes would be formed, they undergo isogamy. If they are of similar shape and size, they would be isogamous and their fusion would be termed as isogamy. Second is the condition when we have anisogamy, where two gametes are of different size. The fungal mycelium has given rise to the gametes and it could be in this form. Then we have oogamy. There is a large sized egg and a motile sperm which comes to the egg and germinates, fertilizes, not germinates, fertilizes, it forms the zygote, it would undergo meiosis and then they would germinate. Then we have spermatium and trichogyne. This is present in the simplest form of fungi, and we do not get into detail of this because then we have to understand other parts as well. You just have to remember that this is also one of the methods in which fungi can reproduce. Then we have another one, that is gametangial contact. Gametangial contact is basically the contact between the male and female sex organs. They are going to come close in the terms of their fungal mycelium. We say that male and female sex organs, they are going to come close. One would be primary mycelium, other would be secondary mycelium, both the primary myceliums of two different strains, and they would give rise to a secondary mycelium which would be heterokaryotic. Now, this heterokaryotic stage would undergo meiosis and give rise to the zygote, and dikaryon would be there, and then zygote would undergo meiosis and then there would be formation of spores which would germinate. So, basically this part is to be remembered, and this part is a depiction of how two different strains of mycelium are going to come in contact with each other, and there would be transfer of nuclei, there would be a dikaryon stage, and then there would be formation of spores afterwards. Now, let us see understand it in other terms as well. See, fungi are not male or female. There won't be any condition that this is male gamete, this is female gamete. There would be positive and negative strain, plus and minus strain would be there. As you can see over here, let us consider this as plus and this minus strain. Both of these hypha are coming in close contact with each other. There is a gametangial contact that takes place. The nuclei have come close to each other, as you can see over here. When hypha of two types meet, they begin to fuse together. This part. This, okay, this is the gametangial contact. The gametes, we are considering these hypha and the nuclei inside it as gametes, though they are not proper egg and sperm, but they are considered as gamete, and that is going to give rise to a dikaryote condition. And after that, there is formation of zygote. Zygote formation takes place after the karyogamic processes, when the nuclei are going to fuse. When the zygote is formed, then there is formation of zygospore. Now this zygospore, inside the zygospore of the gametangial contact which has taken place, what would happen? This zygospore will undergo meiosis now. Now this zygospore is going to give rise to, this is zygosporangium basically. Zygosporangium will have zygospores inside it which would be meiotic, and those meiotic spores would undergo further mitosis to increase their number so that they can germinate and give rise to new mycelium. So this is all about the sexual and asexual reproduction of fungi. Next, what we are going to study is on the basis of this sexual and asexual reproduction that we have come to know occurs in fungi, what classes are being what class differentiation we have achieved out of this understanding. That is, we are going to see the various phyla which are present in the kingdom fungi.